University, where he met the love of his life and married her a year after. Had a lot less gray hair then. He obtained his PhD in pharmacology from the University of Iowa, had one baby at that point. He then moved to Washington University in St. Louis to do a postdoc with Dr. David Holtzman, where he worked on the role of microglia in innate immune cells of the brain in Alzheimer's disease. In the interim, he had a couple of kids, cats, slightly older than me. He joined the faculty of Washington University in the Department of Neurology in 2015 and is currently an associate professor where he continues to work with Dr. Holtzman to study the role of APOE and immune system in Alzheimer's disease. He continues to be married to the love of his life against all odds. He has 3 kids, which we will be sitting still and very good this entire time. And
get that introduction everywhere I go, no matter who's doing it. Okay, so thanks for the opportunity to come talk to you about the work we do in the lab at Washington University. So, as Dr. said, I work on Alzheimer's disease, and I gave the title about a month ago. Drugs. It's going to be a little lighter on the drugs. We can touch on the drugs. Hopefully, it's an interesting story that I'll tell you a little bit. Here. There we go. Okay, just disclosure of interest. Close. Okay, so Alzheimer's disease was first described in 1906, and it was described in this woman here, Augusta Dieter, who was at the university, or was at a hospital in Frankfurt, Germany. And it was described by a team of researchers working at the University of Munich at the time, Alzheimer. This guy seated here, for whom the disease is named, is her primary physician. And I always like to point out, working alongside Dr. Alzheimer was this gentleman, Solomon Fuller, who was actually a Liberian individual. He was the grandson of slaves from Virginia who had emigrated to Liberia. He went on to become the chair of neurology and also. These 2 gentlemen were some of the first to describe what would become. And Augusta Dieter was a woman in Germany. She was very young, time to develop what we think of as Alzheimer's, in her 50s. Started having cognitive decline, and at the time it was considered a mental health disease. Basically sent to a sanitarium. That is where Dr. Alzheimer saw her. And during the interviews, in his notes, he would ask her questions like, "What's your name? Where are you from?" And the reply was always, "I've lost myself." It's a very poignant description of what happens in Alzheimer's. Sort of your sense of self, your memories, your self, right? And so when she passed away, her brain was donated to Dr. Alzheimer in this clinic, and they made the first descriptions of what today remain the hallmarks of Alzheimer's knowledge. So this is the extracellular deposition of what became known as amyloid plaques, and these intraneuronal inclusions of what became known as neurofibrillary tangles. These are basically insoluble proteins that aggregate in the brain and lead to this disease. And along with that, they noted the appearance of what are known as glial cells. In the brain, you have neurons, you have other cell types, which are together in those astrocytes, microglia, and astrocytes. And they noted the presence of those around the amyloid plaques, which is a hallmark feature of Alzheimer's disease. So today we know obviously a lot more than what was known then. They didn't know what any of these inclusions were composed of. Today we know that they consist of extracellular amyloid plaques, which are primarily composed of a peptide called amyloid beta. And these are short peptides, about 40 to 43 long, and they form these insoluble amyloid-like formations. So they form heated beta sheets. They just kind of stick together and they become insoluble and crash out. And surrounding those are different cells. One of the pronounced cells that will surround an amyloid plaque is called a microglia. And these are immune cells in the brain. They're basically a macrophage. So they're part of the innate immune system. And it's been kind of a mystery what they were doing when they surrounded the plaque. Some people thought maybe they were promoting inflammation around the plaque, leading to degeneration. Over the past 10 years or so, we've learned that they probably actually serve a protective role, and they're responding to the plaque to limit the damage the plaque is causing. And another hallmark of Alzheimer's disease, as I mentioned, is the tau pathology. So tau is a protein that's expressed mostly in neurons, and it binds to microtubules in the brain and sort of helps their generally. Well, what can happen is it can also misfold, and it does that in a few different ways within. So it can form what's called a neurofilary tangle, and this is a formation of protein that occurs within basically the cell body of the neuron. It can also perform. It can also form what's called a neuritic plaque tau inclusion. So this occurs within axonal segments of the neuron that are adjacent to neuritic plaques, these amyloid plaques, and it basically will sit in this swollen axon and aggregate there. And these aggregates of tau are hyperphosphorylated, whether that promotes their aggregation or is a result of their aggregation. That's what we see. The other, of course, major component of this is that you have significant neuronal death. So this is a normal, healthy brain individual, and then this is the brain. You can see that there is lack of brain region here called the hippocampus, which is for learning and memory, that gets severely atrophied in Alzheimer's disease. There's significant thinning of the cortex compared to control, then this enlargement of the ventral. So the question is, and was for basically 100 years, was how all of these things fit together. So what came first in all these things? Were the amyloid plaques responsible? Was the tau responsible? What was the role of this relation around the plaques? And what was the sequence of events in which this all occurred? So basically, to cut through about 50 years of Alzheimer's research, what we now know is that amyloid deposition is probably the earliest detectable event that occurs in Alzheimer's. So about 15 to 20 years before you would become cognitively impaired, you begin to develop amyloid plaques. That's denoted by this. So these will begin to accumulate, and then about 20 years after that occurs, what happens is have the appearance of neurofibrillary tangles in your cortical. This is what closely corresponds then with the neuronal death and brain atrophy that is associated with memory loss and losing. And along the way, then you also have the activation or and neuroinflammation that sort of increases along with the pathology and with the pathology. Another important component that we've learned over the past 10 or 15 years is that tau pathology spreads from neuron to neuron and sort of template its own message. So if you're aware of things like prion diseases, if you're deer hunters, there's chronic wasting disease, deer. This is where a protein can become misfolded, go into another neighboring cell, and cause perfectly normal protein to also misfold in the same. This is how we think that tau is then spreading in the brain. So within everyone, probably over the age of 50, there is sort of relatively benign tauopathy, like primary age-related. This is just, it will look like a neurofibrillary tangle, but it doesn't really have. Individuals that have going to develop Alzheimer's, they will have amyloid pathology in the cortical region here that has developed over the. Then at some point, tau pathology will spread from this parahippocampal gyrus into the cortical region. This is what causes symptoms that we associate. So genetics play a huge role in who's going to get Alzheimer's disease. The biggest risk factor for late onset Alzheimer's disease, which is more APOE4. APOE is a lipoprotein in your brain. You might be familiar with it in terms of your cholesterol metabolism in your body, but your brain makes its own supply of APOE. And it's mainly made by astrocytes in your brain. And folks who have an APOE4 allele, one allele will put you at a 4-fold higher risk. And if you are misfortunate enough to have 2 alleles, you're at a 12-fold higher risk of developing. So this E4 genotype kind of made the news. A few years ago, there was an actor doing like a show. Anyway, he did his 23andMe genotyping thing, and like, oh, let's go through your genetics. They saw he was E4. So that was the E4 covered. There's also very rare genetic and cause Alzheimer's. These occur in enzymes that create A beta, PSEN1 and PSEN2. So these are part of an enzyme complex which goes along and cleaves protein that cuts up APP into the little A beta peptides. There are variants both in APP and in that enzyme complex cause Alzheimer's and autosomal dominant. What do we mean by autosomal dominant? Everyone, of course, has copies of genes. Autosomal dominant mutation is one that you have that mutation. Phenotype associated with that mutation will. So a recessive mutation, you would have to have both copies with that mutation, dominant. So basically, if you have a parent who would have autosomal dominant Alzheimer's, you have a 50% chance of developing that. These are extremely rare. They account for less than 1% of all Alzheimer's cases. Again, they're caused by mutations. Basically, all of them are caused by mutations in genes. And these mutations basically serve to increase the production form of A beta that really prone to make a whole bunch of this aggregation prone A beta, wind up getting amyloid plaques in your brain, and wind up dementing sometime in your 40s. So there's a couple different groups that are devoted to studying autosomal dominant Alzheimer's disease. Poses a great way of trying to understand how the disease progresses late onset as well. And there's a very unique group of these autosomal dominant AD patients, Colombia, and they're known as the Antioquia. And it's in this one, they call it the department, basically state in Colombia, called Antioquia, and particularly within this extremely mountainous region. And it was first described by a physician in Colombia who recently passed away just 2 weeks ago named Francisco. And he was a physician in Medellin, in Colombia, and he would have people come from this rural area, and they would all get demented in their 40s, get Alzheimer's. And so he collaborated with some researchers in the United States, beginning in the 90s, maybe in the 80s, and they genotyped these people, and they discovered that it was caused by a mutation in presenilin-1, the enzyme that makes A beta. And there was this glutamate to alanine mutation. And because this region was so geographically isolated, this mutation just kind of mutated. And going through a lot of neurological records and anthropology, they were basically able to trace this mutation to a single Spanish individual from the 60s who immigrated to Colombia, probably say he was came to this region, and now there's more than 25 extended family groups descended from this one individual who carry this mutation. So these folks are destined to develop Alzheimer's disease around 45 years of age. There's 6,000 people in these families, about 1,500 of which currently carry that mutation. So this is the largest single autosomal dominant Alzheimer's kindred. Now, a very provocative paper came out in 2019 that there was a woman who was positive for carrying that precedent could have developed Alzheimer's at 45. And she passed away basically perfectly fine at the age of 70. So she never developed Alzheimer's. But what she did have, when they went through and they sequenced her genome, trying to figure out why didn't this woman develop Alzheimer's? And they found another very rare mutation called the APOE Christchurch mutation, and this is what this is. She was homozygous for this exceptionally rare APOE allele. And given the risk effect of APOE that I mentioned earlier, I thought this was important. So this woman, we have different ways now of measuring amyloid or tau pathology in the brain of living people. So it used to be only be diagnosed accurately at death, and in the past 30 years, whatever, we've developed biomarkers and fluids and different PET imaging methods that we can now tell who has Alzheimer's or who will develop Alzheimer's. One of these methods is to use what's called PET imaging. So we basically have radioactive ligands that we put in, and we can see where the amyloid plaque is. So you can see here these red regions in this woman's brain are where she had a amount of amyloid plaque. This is a standard presenilin 1 mutation carrier at age 45 when they get Alzheimer's disease. You can see there's definitely positive here, different brain regions, cortex. She had just a massive amount of, even above that, probably because she lived an additional 30 years cranking out A beta, positing plaques. But what was different here is that compared to autosomal dominant cases where you could see this here is their measuring their tau pathology. So it's another PET ligand. Where you see red here, that is where they have tau tangles. Those are the brain regions that are degenerating. You can see a standard autosomal dominant patient. They have lots of tau pathology in their brain, corresponds to the symptoms. This woman was basically protected from tau pathology, even up to 70 years. Head full of amyloid, no tau. So how could this have happened? So this led to the hypothesis that somehow Christchurch was reducing the spread of tau into brain regions that had pathology. So what is the APOE Christchurch mutation? It was discovered in Christchurch, New Zealand, but it's an arginine to serine mutation. So here's a nice bar diagram of APOE. So it has a receptor binding region here that binds to different receptors in cells to mediate APOE uptake. And the three common isoforms of APOE are APOE2, APOE3, and I mentioned that APOE4 is that risk variant that increases your risk. So 1 in 4 individuals has an APOE4 allele. 50% of people with Alzheimer's have a very profound effect on who develops. APOE3 is the most common isoform of APOE. About 80% of people have one of those. And APOE2 is actually a protective allele. So it's very rare. It's more rare. 1 in 10 people have an APOE2 allele. But having one copy will cut your risk by about half of developing Alzheimer's. What's kind of interesting biologically are these are all sort of discriminated by a single amino acid. We're still working to understand how these single amino acid changes in cysteine and arginine affect how this protein affects Alzheimer's disease. But it's a very profound. In contrast to these, Christchurch mutation is globally present in about 1 in 16,000. It's a very rare mutation. And this woman had 2 copies of those, so that's just like her. Come to find out in reading about this mutation, it's actually might be slightly enriched in Spanish populations. So maybe someone else who came along with the vanilla mutation was apparently Christchurch. Now, mix a little geographically isolated there. Okay, so how are we going to study this hypothesis that this mutation in APOE is affecting this one? So we have a few different, most of them involve mice. So we have mice that can be genetically engineered to develop amyloid pathology. You can overexpress human APP and human PS1 that contain mutations that cause Alzheimer's disease. To basically make these mice develop it. Mice won't develop it on their own. You have to give it to them. And so if you take these mice, they express these transgenes, and then over however many months, depending on the model, they will develop more and more amyloid. The amyloid pathology they develop looks remarkably similar to the type that's here in the mouse. The amyloid's blue, and then you have microglia, white stain. And then this is the human brain, way to this bluish purple here. So the microglia are we also have mice that develop tau pathology. They can overexpress human tau containing mutations that don't cause Alzheimer's disease. They cause other diseases that are called tau that generate different parts of the brain. But they will develop what look like neurofibrillary tangles and get brain degeneration. Another way that we can model the disease in mice is to take those amyloid. And if you take a human AD brain and you do a biochemical isolation, you can isolate out tau fibrils from that brain. And then, since tau can template its own misfolding, you can inject those into an amyloid mouse. The mouse will develop dendritic plaque tau pathology around the amyloid plaques. Again, that looks very similar to that. And a final tool we have is we can what's lock-in mice. So we can take mice and we can manipulate their genes to express human. So we have a whole set of mice in which we've taken out the mouse APOE. We've inserted different human APOE. These mice, APOE2, APOE4, APOE3. So in our case, we elected to make a mouse that expressed human APOE3, the Christchurch mutation. And given that the woman developed a lot of amyloid pathology, we crossed those mice to amyloid pathology. Then we decided to use this tau-seeking approach, inject tau fibrils into the brain, and see if the presence of the Christchurch allele, the mouse developed a lot or a little pathology around the plaque. So this study was led by a graduate student in the lab, Chen. And again, he developed 2 strains of mice here. So an amyloid mouse expressed human APOE3 or human APOE3 Christchurch. And at about 6 months of age, these mice have a pretty good amount of amyloid pathology. So mice won't develop tau pathology on their own. You have to give it to them. So he injected AD tau from AD brain, injected it into the mouth of the cortex. And then he aged it another 3.5 months, that pathology time to spread in the brain. And then he looked and see what he saw. So the first thing, here's the mouse brain. And this is a mouse brain stained for A beta plaque. So you can see in the APOE3, you have a certain amount of plaque. It's all cortex region here. And the campus region here and here. But this was really reduced in the APOE3 Christchurch and the quantification. But this was a little different and caught us off guard at first. We didn't necessarily expect this because the woman had far more amyloid. But you have to again remember that she was very old when they did that PET imaging. So if they had imaged her maybe at 45, she might have had less or less amyloid. But in terms of the p-tau, so we antibody against phosphorylated tau, and we stain these mouse brains. And you can see that this sort of gray dots here in circles, that's phosphorylated tau around the plaque, right? So that's that dendritic plaque. And that was strongly reduced in the APOE3 Christchurch. So this kind of mimicked what we saw in the patient. Now you might say, well, she had less plaque, so she might have less tau, and that's just. So we next did some fluorescent imaging, and we just looked around it. We said, okay, we look at any given plaque, is there less tau around? The plaque here is in blue, the phosphorylated tau is in. You can see in the APOE3, there's a certain amount in hippocampus and cortex, and this is again strongly reduced when you have APOE3 Christchurch, mimicking what we saw. So how could this be happening? So over the past 5 or 10 years, prior to doing this work, we've been working on the idea that microglia can mitigate the ability of amyloid to cause tau pathology. And this was due to a number of different genetic risk factors to get into. But the crux of the matter is that microglia around plaques appear to limit the ability of amyloid to cause this zonal blebbing that can be the site for tau pathology would aggregate. And that's either through clearing away these aggregations or plowing the amyloid to where it doesn't have physical contact with parts of it. So we said, okay, we're seeing less p-tau pathology, plaques. Maybe this is happening through the so we did a stain. So IBA1 will label all the microglia, all their cell bodies. Then CD68 here is a marker for microglial activation, where microglia are ramping up their lysosomes, sort of digest things they're taking. So in human APOE3, you can see very nice clustering around the plaque, but this is even a bit more exaggerated with the APOE3 Christchurch. So you're getting a much stronger immune reaction. And along with that stronger immune reaction, you not only have more cells, but you also have more of this CD68. So the lysosomes are working even harder than they are just have normal. So now we got to step back and we say, okay, so what does this have to do with tau getting into the neurons so you have more microglia? Well, tau gets in through, it's not entirely clear how tau is getting into it's in the extracellular space. How's it getting into the neuron to cause this misfolding? There's a couple different receptors. One is called LRP1, and LRP1 partners the glycans on the cell surface take tau from the extracellular space inside the cell. So this is receptor-mediated endocytosis. It could happen in neurons, could happen in microglia. If it happens in, say, a microglia, it could go through into the endolysosome, and then wind up in the lysosome and get degraded, and your tau goes away. Good deal. It could also happen that you maybe take it up into a neuron, and it leaks out of the lysosome, and then it can cause misfolding of tau and propagate it, right? One other aspect of this is that these receptors, LRP1 and HSP3, are also receptors for this got us to thinking, well, maybe APOE and misfolded tau are competing for the same receptor. And so maybe APOE3 Christchurch doesn't bind as well. And in that original paper, they'd actually looked at the ability of APOE Christchurch to bind HSP. So they ran a column here, and basically they loaded a bunch of APOE onto a heparin column. And then they ran increasing concentrations of sodium chloride through the column. So more tightly it's bound, more concentrated your sodium chloride is then wash it off. Now, APOE Christchurch came right off, so it bound very weakly to the column, especially when you look at, say, APOE 2, 3, or 4, so you really had to crank this thing full of sodium chloride to get it off. So you had decreased affinity, right, for APOE3 Christchurch with this receptor that tau also has. That means it's not really going to compete as well for tau. So microglia will be able to just take up tau through that receptor all day long and degrade. At least that's what we thought. So we decided to test this. We took cells, we cultured macrophages, which are very similar to microglia. And we took tau and we labeled it with a fluorescent dye so we could see. Took that tau, we put it on the cells, and then we did flow cytometry to look at which cells were green and which cells were taking up tau. We did that with 2 different concentrations of this fibrillar tau. We saw that the APOE3 Christchurch, which are these triangles here, took up a lot more of that fibrillar tau. So these cells were able to take up more of this stuff in the extracellular space. So we said, well, is this going through these receptors? So there's different inhibitors you can use to block HSPGs. Rat protein here will block LRP1. And when we applied those to the cells, found that blocking those receptors reduced the tau levels. That kind of suggested that these receptors. Then we said, well, what happens if we just dump in a bunch of APOE? And we outcompete that tau. And so if we took APOE3 cells and we added a bunch of APOE3, if we added APOE3 Christchurch, it went up a little bit, but it wasn't necessarily that different. But if we took APOE3 Christchurch cells and we added APOE3, then we were able to block that tau from getting. But if we added a bunch of APOE3 Christchurch, it didn't have that same effect because you had that reduced effect. This led us to think that the ability of microglia to take up tau is improved with the Christchurch mutation because Christchurch wasn't interfering with the same effect. So they're taking up more, but are they able to degrade it? And so we did a degradation assay where basically we took cells, APOE3 or Christchurch fluorescent cells. We added again that fluorescent tau, and we just let it sit. And then we looked for 12 hours, 24 hours, 48 hours later, and we saw how much. We saw again the Christchurch were able to take up a lot more. By the time you get down here to 48 hours, they've been able to degrade all of them. There's just a lot less time. They're a lot more efficient at degrading. And the final thing that a cell can do is it sometimes just release undigested vomits it out. So we looked at that, and we saw that APOE Christchurch myeloid cells were able to digest more efficiently compared to the APOE3. APOE3 will spit back out a lot more of that fibrillar tau that can then go on to spread throughout all the different cells. So to summarize what I just told you, we have insoluble tau fibrils. Those can be taken up through receptors shared with APOE. And if you have APOE3 Christchurch, it doesn't bind to those receptors, so that tau can get taken up and degraded a lot more efficiently. And in theory, then, that increased microgliosis around the plaques, that increased microglial clearance of tau, is going to protect the neurons around the plaques, developing that tau pathology. That's going to prevent the tau pathology from spreading through the brain, causing the neurodegeneration. So how can we leverage this as a therapy? Well, if you want APOE to stop interfering with the ability of these cells to cure tau, you could just reduce the amount of APOE. And so there's different ways you can do this. In the past, we've looked at using tools called antisense oligonucleotides. They're basically little RNAs that you can put in, to DNAs that you can put in, and they'll lead to the degradation of the RNA target. So we can decrease APOE expression in the brain using those. Other groups are looking at generating antibodies that will sort of mimic the effect of APOE Christchurch and giving those. So I was in college, kind of want to know what you want to do with your lives. So there's a ton of different career paths in Alzheimer's, or really any biomedical research you do. So some of the people we interact with on a day-to-day basis are, of course, physicians and bench research, medicinal chemists, but also statisticians, engineers, a lot of equipment used in these assays. There's a huge push for more bioinformaticians. These are people that work with data sets, basically. Then on the more clinical aspect, the Alzheimer's Disease Research Center at WashU, there are social workers that work with patients directly, clinical research coordinators that work on patient outreach, serve community outreach, and a huge push for that. There are science writers. These are people that write up press releases or memos to give to donors, things like that. So that you understand the science and then put it in words. If you have a great idea, you need to get it from the lab to the clinic. You need to have a company at some point. So if there are patent lawyers, business development people, and then people to help lobbyists. The Alzheimer's Association is probably one of the most impressive medical lobbyist organizations out there. There's earmarked money in the federal budget every year just for Alzheimer's disease. And foundation relation officers. These are people at universities or foundations that solicit donors. They're able to explain science to wealthy, interested parties. So, final group here, the Holtzman lab at WashU. There's about 30 of us or so. Dave Holtzman, so he's a great guy. He's the stepped down as chair of neurology a couple years ago now. Yeah, and we've had a few Maryville students come through and work with us. Yeah, I'll take any questions. Yeah.
Stop. What
Right. Yeah, no, excellent question. So if you have a symptomatic patient, it's very hard to sort of bring the brain back. So there's a huge push. I've seen this analogy before. This was there were so many failed amyloid drugs. So now currently there are 2 amyloid antibodies. MB and donanemab, I can't. Name is. Previously, they would give these antibodies, and none of them would work. They're like, oh, this sucks. But they were giving them this stage of the disease, where the amyloid has been there forever and sort of already degenerated. And it's sort of like trying to take your kitchen after your sink has been overflowing for 3 weeks. The kitchen looks like this. And you say, why does my kitchen look like this? I turn the sink off. What's done is done. If you try to go preclinically and you were to say lower tau, that's actually in the clinical trial right now. So theoretically, that will prevent the dementia. And the same thing with the theoretically, if you strengthen the microglial response around the plaque, that would prevent the dementia from happening. So that's kind of the goal, is to stop it from happening. So then.
Good question.
Anything at all. Yeah.
That's looking at improving some of the house cleaning capabilities of the brain.
House cleaning? Yes.
So it's
Yeah.
every night.
So yeah, what Helford's referring to is sort of this lymphatic clearance. So over the past 10 years ago, 10 years, 10 years for everything, people discovered that brain tends to sort of clean itself, right? So when you sleep, you have more, theoretically, more chance for the A beta peptide to clear out. So there are different ways of promoting that. Folks are working on promoting lymphatic drainage from the brain using different growth factors. And then there's a huge wing of the Holtzman lab that works on this. And kind of what they found was that slow wave sleep is especially potent at drainage from the brain. Metabolic background, right? So A beta or tau or whatever. So there are different sleep drugs that are especially good at promoting slow wave sleep. Lemborexant, I think, is like an orexin receptor agonist. Okay. It's a good drug. It helps you sleep and promotes slow wave sleep and the clearance. So that's another sort of lifestyle pharmacological way that people are working.
slowly. So like, it's so
So there's a woman at Boston, who like the world's expert at chronic traumatic encephalopathy, what a lot of those injuries wind up in. And basically, they don't quite develop Alzheimer's disease, but what they develop is a different type of tau brain. Develops in a different place, but the outcome is kind of the same. They develop cognitive dysfunctions, personality changes, depressions. So there's something to do with repetitive brain injury associated with athletics or other things that can happen. But you typically think of the NFL players developing disease, that leads to this aggregation of. So there's a few proteins in the brain that, for whatever reasons, prone to aggregation. Tau is one of them. ALS caused by a lot of protein aggregation. Then there are other.
Makes you want to go to this field of research.
Yeah.
So now you're called. So what type of research appealing to you?
Yeah. I think most people that get into Alzheimer's research, if you catch hard enough, you always find that they have a personal connection. So for me, it was my grandpa had Alzheimer's disease, and so I kind of got interested in it that way. I think a lot of people in the group can say the same thing, had a grandparent or an aunt or a parent. All right, thank you guys.